Now we move to our next speaker, Dr. Chauhan. Sir is professor and head Department of Biotechnology, All India Institute of Medical Sciences, New Delhi. Dr. Chauhan has received his PhD in biochemistry from VP Chess Institute, University of Delhi. Sir has worked as postdoc fellow at University of Pennsylvania School of Medicine, Philadelphia, and at National Cancer Institute, Bethesda, USA. Sir has joined AIM during 1992 as Assistant Professor, Department of Biochemistry. Sir has been honored with many prestigious awards like Dr. V.K. Khanolkar Oration Awards of National Academy of Medicine Sciences in 2021, AIM Excellence Research Award for 2 consecutive years, and have many more awards to his credit. Sir has authored more than 110 original research papers in index peer-reviewed journal and invited review and chapters in book. Now I invite Sir for his talk.
So what I'm going to talk about is association of human cathepsin L promoter hypermethylation with blast crisis and resistance to chemotherapy. This is, as Dr. Mithu Banerjee told, that this is one type of epigenetic change that takes place in the promoter region. So I'll start by a very brief introduction about leukemia, although all of you know it is made up of 2 words, leuka and emia, which means white blood. Essentially, it's a malignant progressive disease in which the bone marrow and other blood-forming organs produced increased number of immature abnormal leukocytes, and they suppress the production of normal blood cells. This is a blood picture of a leukemic patient. You see a lot more number of white blood cells, mature or immature, as compared to the normal individual. And these are some of the common symptoms that are observed in leukemia patients, which are systemic as well as organ specific. It includes weight loss, fever, frequent infection, shortness of breath, muscular weakness, pain or tenderness in joints and bones, fatigue, lymph node swelling, spleen or liver enlargement, night sweat, etc. And this list is by no means complete. Now, this is a picture of how these progenitor cells, hematopoietic stem cells, differentiate into lymphoid or myeloid progenitor cells. And then, depending upon where the problem is, this leukemia is categorized as myeloid leukemia or lymphoid leukemia, depending upon the origin of the progenitor cell. And depending upon its onset and how quickly it develops and what are the symptoms, it is known as chronic or acute. And what I'm going to talk about today is chronic myeloid leukemia, which is of myeloid origin. It develops over a period of time. Symptoms are very mild, and it may go unnoticed for a while, except when it progresses into accelerated phase and blast crisis. And as it progresses, the survival of the patient reduces. That has been shown in accelerated phase. It used to be only 12 to 18 months. Now it is 4 to 5 years. And in blast crisis, now it is only 6 to 12 months. So now I'm going to switch gear and tell you a little bit about cathepsin L, the protease on which we work. And before talking about cathepsin L, let me tell you about cathepsins. Cathepsins are a diverse group of lysosomal proteases which are found in all organs. There are about 11 known cysteine cathepsins. And why do we call them cysteine cathepsins? Because there is a cysteine amino acid which is present at the active site, and that is essential for the catalytic activity. They perform several essential cellular functions, which may include protein turnover and degradation, antigen processing, hormone activation, lysosomal cell death, autophagy, and aging, as well as cell survival. And then again, this list is by no means complete. My laboratory has been working on 2 such proteases, which are cathepsin B and cathepsin L. And But I'm going to present some of the results on cathepsin L in CML. Now, as I told, cathepsin L is localized in the lysosomal compartment. And if due to the injury of lysosomal compartment, if it is released into the cell, then it causes cell death. And that's why, so cell deals with it with the help of endogenous inhibitors, which are known as cystatins, which have been shown here in the form of triangles. So several years back, we demonstrated that cathepsin L is overexpressed in a number of human cancers. And this is the list of human cancers, and its expression was found to be 10 to 50-fold higher in cancers as compared to the normal cells. Now, majority of the cathepsin L synthesized by the malignantly transformed cells is secreted, and this is a slightly complicated slide, and we demonstrated that its secretion, for secretion to take place, its carboxy terminal plays a very important role. But I want to draw your attention to the fact that KB1 cell is a tumor cell, epidermoid carcinoma cell, and you can see in the cell very little of it is present in the cell lysate, and you see a thick band in the culture media, suggesting that about 90 to 95% of it is secreted into the culture media. And using the information that we had published, that its carboxy terminus is important, another group working in the same area, led by Raymond Fred in France, demonstrated that if you block the secretion of this protease in human melanoma cells by using single-chain variable fragment of cathepsin L against the carboxy terminal, then and inject these cells in the nude mice, then the size, tumor diameter significantly reduces. As you can see here, these curves in the bottom, whereas the tumor size nicely grows, thereby suggesting that if you block the secretion, it abrogates the tumorogenicity of melanoma cells, which are known to form large-sized tumor in nude mice. Similarly, if you block the secretion, it also abrogates the invasive ability of these cells, and also they abolish the metastatic potential. This experiment was done by injecting these cells where secretion was blocked into the tail vein of nude mice, and nodules formed in the lungs were counted. And as you can appreciate in these slides, parental cell line, or which were transfected with neomycin resistance gene alone, you see about 100 to 200 to about 160 tumors in different mice that were injected with this. But when you block the secretion, the nodules are decreased to 4 to 40, thereby suggesting that even the metastatic potential is abolished. This is the schematic representation of cathepsin L gene, which we cloned several years back. It consists of 9 exons and 8 introns, and it is transcribed by 2 different promoters into multiple splice variants, which are translated with different efficiencies. These are our publications, and I'm not going to talk about this today. So there were about several papers. So one thing that we noticed while working on cathepsin L gene, that it is localized on chromosome 9q21.22 locus. And that was published in 1993, much before the human genome sequence was available. Even in human genome sequence, we see localization of cathepsin L gene on chromosome 9q21.22. And if you remember, or those who do not work on CML, I would like to draw your attention to the fact that in CML is characterized by Philadelphia chromosome, which is formed due to the reciprocal translocation of long arm of chromosome 9 and 22. And this is close to the locus on chromosome 9 where cathepsin L gene is present. So what it does, it results to the formation of a fusion gene between BCR and ABL. And this BCR-ABL is a constitutively active tyrosine kinase. And if you express BCR-ABL chimeric gene in any hematopoietic cell, it can lead to leukemia or CML. So this is very essential. In about 95% of the patients, you are able to detect BCR-ABL at any stage. So with this information, we asked ourselves two questions. First question was, does this chromosomal translocation has any effect on cathepsin L expression? Because it is very close to the locus where BCR-ABL tyrosine kinase is formed. Also, can this alteration in expression of this protease have any prognostic, diagnostic, or therapeutic significance in CML? We tried to answer these questions through the following experimental design, which I am going to share with you this afternoon. So to address this question, we recruited about 47 CML patients and also 30 patients with systemic infection, because the number of WBC counts are very high in CML patients, and WBC counts are also high in systemic infection. Also, we recruited 50 healthy volunteers, and this study was undertaken after ethical clearance from our institutional committee. We collected about 10 ml of peripheral blood and isolated mononuclear cells from these patients as well as controls by 5-fold density gradient, and we assayed the following. What we assayed is we assayed cathepsin L expression by quantitative real-time PCR, enzymatic assay, and Western blot. And in addition to this, we also assayed BCR-ABL mRNA levels as well as VEGF levels. VEGF levels because VEGF is known to be overexpressed in CML patients. That's why it was used as a positive control. And then we simultaneously isolated DNA from these cells and studied epigenetic changes in the cathepsin promoter. And I'll tell you the reason for doing this subsequently. And these are the baseline characteristics of CML patients. And I want to draw your attention to the fact that this is the leukocyte count in normal controls, which was higher in systemic, in patient controls with systemic infection. And these are CML patients. So out of these 48 patients, 47 patients, 31 were in chronic phase and 9 were in accelerated phase or blast crisis. So we assayed cathepsin L activity in these individuals, and to our surprise, cathepsin L activity was significantly higher, about 4 to 5-fold higher in CML CP patient, but it was only about 1.2-fold higher in accelerated phase for blast crisis. So this was kind of surprising. Same thing was observed in case of cathepsin L mRNA, which you can appreciate here on the right-hand side. In blast crisis or accelerated phase patient, it was higher, but not as high as in chronic phase patients. This is a cell line which was established using from a blast crisis patient or accelerated phase patient, and we used in all our experiments as a positive control. So activity of cathepsin L and mRNA were comparable in K562 cells and accelerated phase and blast crisis patients. There was a direct correlation between cathepsin L mRNA and activity as well as protein levels. We also performed Western blot, which has been shown here, and you can appreciate the same change in cathepsin L protein levels in chronic phase patient compared to the accelerated phase and blast crisis. Three bands are evident in the Western blot because it is synthesized as a pre-pro cathepsinel, which is converted by autoactivation into pro-cathepsinel and then finally into its active form. This was a tubulin control and there was no difference in the tubulin level, thereby confirming that cathepsinel is significantly higher in case of chronic phase. It is higher in blast crisis also, but significantly lower than the chronic phase. We also assessed the expression of its endogenous inhibitor, which is cystatin C, and as expected, even the endogenous inhibitor was significantly lower in case of CML-CP patient and blast crisis patient as compared to the patient control and healthy individual, and there was an inverse correlation between cathepsinel activity and cystatin. So we also assayed PCR-ABL expression, as I alluded to you in the beginning while discussing about the experimental design, and VEGF levels. And what we found is that there was a direct correlation between PCR-ABL level, very strong correlation between PCR-ABL level and VEGF mRNA, PCR-ABL and cathepsinel mRNA, but no such correlation was observed in the blast crisis or accelerated phase patients. None of these. This was insignificant. So that means BCR-ABL or VEGF did not have any correlation in accelerated phase or blast phase. Then we went ahead and studied if there was any correlation between VEGF and cathepsinel. And to our surprise, we observed strongest possible correlation, and R value was 0.97 between VEGF and cathepsinel mRNA. So suggesting that VEGF is responsible for, there is a cause and effect relationship between cathepsin and VEGF overexpression. To address this issue further, this is relative abundance in VEGF. So we did a very simple and straightforward experiment. We took peripheral blood mononuclear cells from normal healthy individuals and treated it with VEGF. And to our surprise, it resulted in a significant increase in cathepsinel activity upon treatment with VEGF. However, no such effect was seen when we took cells from the patients. And the reason that we could think of is that already there is so much VEGF present in these cells or in these patients that these VEGF receptors are already saturated, and that's why it is not able, additional VEGF is not able to elicit any response in these. To confirm these findings, we did transcriptional regulation studies, and I'm not going to go in the detail. We cloned cathepsinel promoter upstream to eluciferase reporter gene, and then treated these cells with VEGF after transfection with the promoter reporter constructs, and we observed a significant increase in luciferase activity upon VEGF treatment. By doing a series of promoter deletion analysis, we identified VEGF response element in the cathepsinel promoter, but I'm not going to discuss about it. These results have already been published. Also, when we analyzed cathepsinel promoter, we found a CPG island. As Dr. Mithu Banerjee mentioned, a CPG island was found in the cathepsinel promoter, and these are promoter elements which have been shown in red color, and these are enhancer elements shown in yellow color. And this green stretch shows the region of cathepsinel promoter, which starts from the promoter region and extends into the first exon. A number of CPG methylation sites were identified in this, and next slide shows these sites which have been shown in boldface, and there are about 106 methylation sites were identified in this region. About 40 to 42 of them were there in the promoter region. So we looked at the methylation status of these sites, and as has already been told about Dr. Mithu Banerjee in her talk, that we used bisulfite sequencing, and the principle of bisulfite sequencing has already been explained to you, so I'm not going to go into the detail of this. So these are different. So we performed these experiments in 10 representative samples of each group. Accelerated phase, blast crisis, we had only 9 samples in blast crisis, so we could only do in 9. Otherwise, we took 10 samples from each group. And K562 cells, as I mentioned you before, is a blast crisis cell, and that's why we used this as a positive control. I would like to draw your attention to the fact that all these filled circles are methylated methylation sites, and open circles are unmethylated. Now, if you see in chronic phase patients, there were very few sites which are methylated, but in patient controls as well as in normal healthy individuals, these sites were extensively methylated. But as expected, these sites are significantly more methylated in accelerated phase or blast crisis patients. It is significantly more methylated, which was also evident in the expression level of cathepsin L, both at mRNA and protein level. So, thereby suggesting that this lower expression in cathepsin in accelerated phase or blast crisis was due to the methylation. To test our hypothesis, we used demethylating agent, which has already been told to you, 5'-azacitidine, and we did this experiment in K562 cells, which were established from blast crisis patients. And if you see, treatment of these cells with 5'-azacitidine led to a time-dependent increase in cathepsin L mRNA levels. There was no difference in cystatin C levels or 18S ribosomal RNA level, suggesting that this is specific for cathepsin L. So this confirmed that reduced expression in blast crisis was due to the promoter methylation. We saw the same thing at protein level and enzymatic activity. Also, you can see here it's 0, and then this is 48 and 72, and you see how the band is increased. The width of the band increases. This is vehicle-treated control, and so this confirmed that silencing of cathepsin L expression in blast crisis is due to the methylation of the promoter. Also, this goes without saying that prognosis of patient in blast crisis is very, very poor, and survival is only 9 to 12 months. Whereas in chronic phase, it is 5 to 6 years or even more. Now these patients can live longer with the treatment. So that means this suggests an association of promoter methylation with blast crisis or accelerated phase and poor prognosis. Now, we confirmed these experiments in PBM isolated from blast crisis patients. This is patients PBMCs, and this is vehicle treated, and this is treated with 5'-azacitidine. And you can see here, upon treatment with azacitidine in blast crisis patient PBMC, there was a significant increase in cathepsin L mRNA level, its activity, as well as protein level, thereby confirming our results of K562 cells in patients. So then we went ahead and overexpressed these cells in K562 using a heterologous promoter, and then demonstrated that imatinib mesylate, which is used for treating these cells, actually the cell survival reduced significantly in cells which were overexpressing cathepsin L, and thereby suggesting that overexpression of cathepsin L sensitizes these cells, and its reduced expression makes it resistant to this. So this is summary of the treatment, summary of these results. And what I want to say is, I have already told, we observed a decreased expression of cathepsin L, expression in blast crisis, which was due to promoter methylation. And then if we overexpress it, it sensitizes the cells to the treatment which is given in leukemia. And these results were published in Journal of Cellular and Molecular Medicine. This is the this work was done by my PhD students Garima Pandey, Misty Samaya, and Prerna Jain. Dr. Samir Bakshi and Dr. Lalit Kumar from IRCH Cancer Center at AIMS helped us with the samples, and funding in my lab was from DBT, DRST, and DRDO. Thank you very much for your attention.
Thank you, sir. Thank you so much for your informative talk.